at the Stevens Neuroimaging Institute, we take thousands of images of people's brains and attempt to understand why is a brain that has been afflicted by Alzheimer's, why is it different than a normal brain? And how does that change over time, over the course of a person's life?
So we get almost a crowdsourcing effect here. We are involved in more multi-site data acquisition projects than any other place in the world, where we have scientists around the world cooperating, sharing their data, sharing their tools, sharing their results. This is accelerating the pace of discovery beyond anything that has happened before. Okay, so what we're looking at here is this is a cut of a person's brain, and the size of the ventricles here are abnormally large. MR imaging is an important tool in our arsenal of looking at brain. It is perhaps one of the most important tools. Lighter colors are fluid, darker colors are white matter. It's actually where the brain is wired from one region to the other. Now we have 7 Tesla machines that allow us to look at small structures, substructures within the hippocampus, a very important area in Alzheimer's disease. It's unbelievable.
So with really high field MRI, it's almost like a game changer in what you can see in the brain. So if you were in a plane over a city, you could infer what some of the glimmers would likely mean. But it's almost like being there on the ground. You can see your cell-to-cell connections, you can see blood flow in millisecond resolution. And you could even see some of these telltale molecules that are a sign of Alzheimer's with much greater precision, and that often means much earlier.
The scans allow us to look at a person who may be suffering from the disease and doesn't even know it. One of the important things about Alzheimer's disease is we often suffer from this for decades before our cognitive decline becomes apparent. Well, we want to make sure we identify those people in the earliest phases of the disease. These darker spaces here, these are fluid-filled spaces, which are also indications of the fact that this person has lost brain tissue. Paints a picture that was unavailable before. This is a picture of a person that can speak to you while in the scanner. We can ask them to perform tasks. Remember these 10 digits, count backwards by 9s, for example, and then look at the areas of brain that are functionally active while they're doing that.
We can learn a lot from studying the post-mortem tissue and understanding what was going on in that brain at the cellular level. So this 3D microscopy is really kind of progressing the field in a new direction. We can look at the tissue at a deeper level than what they can see at the MRI.
Here we have a section of Alzheimer's brain from a human patient. You can see the hippocampus here.
As amyloid plaques and tangles develop, they affect certain brain regions. And using our microscopy approaches, we can see this at a cellular level where in the brain. And characterizing and identifying each cell type and how they're particularly affected by this disease is a really critical aspect of this work, and really developing treatments that can specifically target those areas. We're trying to integrate between what the MRI can see and what we can see in the microscope. So one of the key aspects is to digitize these slides, digitize this into an Alzheimer's imaging database where we can investigate this using big data informatics and machine learning.
The key to understanding something complicated is data. Big data. Big data that describes small things, big data that describes big things, and you need lots of it. In this system, there's about 25 petabytes of data, and a petabyte is a very big number. And all of that data can be made available to researchers who want to examine characteristics about Alzheimer's disease. There's genetics in here, there's clinical data in here, there's imaging data in here of all types, there's behavioral data. The machines have a capability of finding things that we cannot always appreciate, because a machine can track millions, 10 millions, hundreds of millions of data points at the same time. The machine has no limit, basically. We can't do that as human beings.
Imagine if you had a room full of 60 radiologists and all of them were reading your scan. A lot of what artificial intelligence is doing is aggregating multiple sources of expertise, learned novel features, things that we might not in any one individual find the capacity to do.
MRI is one data point. It allows us to look at many facets of the human brain. Those images get put into databases and related to the clinical observations. How well is that person doing in a memory test, for example? We can relate measurements of their cerebral spinal fluid, what proteins are in that fluid. We can relate their genotype, and all of that together is what the machine looks at, not just one measurement. I think that's the power. Putting things back together is essential.
And we think that USC can be it's one of the fountainheads of neuroscience research over the next several decades.
This is a moonshot. You need to take some risks. You need to make investments because we're on the cusp of so many discoveries.